Hello, and welcome back to Genome Medicon. Today, we're exploring a topic that has been a major challenge for health experts around the world for many years: HIV, or the human immunodeficiency virus. With the help of genomics, we are learning more than ever about this virus. Let's see how this new knowledge is helping us to fight HIV. HIV is known for attacking our immune system, making it harder for the body to fight off even common infections. It mainly targets CD4 plus T cells, which play a key role in helping the immune system to do its job. Once the virus gets into the body, it mixes its genetic material with ours, turning our cells into little factories that make more of the virus. This cycle weakens the immune system over time, and if untreated, it can lead to AIDS, or the acquired immunodeficiency syndrome. One of the biggest problems with HIV is how fast it changes. Every time it makes a copy of itself, it can slightly change its genetic makeup. This means that in one affected person, there can be many different versions of the virus. This ability to change so much is one of the main reasons why making a single vaccine that works for everyone is so hard. Scientists have been working for decades to make a vaccine for HIV. Early efforts focused on getting the immune system to create neutralizing antibodies. These are proteins that latch onto the virus and stop it from infecting cells. But because the virus changes so quickly, these antibodies often don't work on new versions of the virus. That's why researchers started looking into other approaches, like vaccines that activate CD8 plus T cells. These are special immune cells that can find and destroy cells already infected by the virus. While this strategy has shown promise, it also comes with its own set of challenges. Another idea has been to use a combination of vaccines to activate multiple parts of the immune system. For example, the prime boost approach uses one vaccine to prepare the immune system and another to strengthen the response. This method was used in a big trial called RV144. It showed only a little bit of success in protecting people from HIV, but it gave scientists important clues about how to improve vaccines in the future. This point of time is where genomics comes in, and it's already changing the game. Genomics is the study of genetic material, and it helps scientists learn more about how HIV works. By studying the genetic code of HIV from people around the world, researchers have created a detailed map of how the virus changes over time and in different places. This information is key to figuring out how to stop the virus. One cool thing that the researchers figured out was that genomics doesn't just tell us what the virus looks like now. It also helps predict how it might change in the future. Scientists have identified parts of the virus that don't change much, even when it mutates. These stable parts are great targets for new treatments and vaccines because they work across many different versions of the virus. One of the biggest discoveries in this area is something called broadly neutralizing antibodies. These are special antibodies that can target parts of the virus that don't change much. Scientists are now trying to figure out how to create vaccines that can make our bodies produce these powerful antibodies. This is still a work in progress, but it's a very promising direction. Recently, new technologies like mRNA vaccines have opened up exciting possibilities. You might know about mRNA vaccines because they were used to fight COVID-19. These vaccines teach our cells to make proteins that trigger an immune response. Now, scientists are using the same method to create mRNA vaccines for HIV. This approach is flexible and can be updated to keep up with the virus as it changes. Another new idea is using mosaic antigens. These are specially designed proteins that combine genetic material from many different versions of HIV. Mosaic antigens aim to train the immune system to recognize a wide variety of virus types, offering better protection. This approach has worked well in animal studies and is now being tested in humans. Before testing vaccines on people, scientists use animals, especially rhesus macaques, to learn how well a vaccine works. These studies helps us see how the immune system responds to different vaccines and how safe they are. In these studies, researchers use a special type of virus called SHIV, which is similar to HIV but adapted for monkeys. These experiments have given us a lot of helpful information and have guided human vaccine trials. Genomics is also helping us to understand why some vaccine trials work better than others. By studying the virus in people who get infected despite being vaccinated, scientists can figure out what makes certain strains harder to stop. This information helps them design better vaccines. Genomics is also leading new treatments. For example, tools like CRISPR, which can edit genes, are being explored as a way to remove HIV from infected cells. This approach is still very new, but it could open up a whole new way to fight the virus. Even with all these advancements, there are still challenges. HIV can hide in certain parts of the body called reservoirs, where it's hard to reach. This makes it tough to completely get rid of the virus. But every new discovery brings us closer to solving these problems. The progress we made so far shows that a world without HIV is possible. Genomics has given us powerful tools to understand the virus and find new ways to fight it. While there's still work to do, the future looks brighter than ever. Thank you for watching Genomedicon. If you found this episode interesting, don't forget to subscribe and hit the bell icon to stay updated on the latest in medical genomics. Until next time, keep exploring the wonders of science and take care.